Hello, my name is Shannon, and I'm a master's student at Northern Arizona University in Flagstaff, Arizona, and I've been working with the USGS in Flagstaff for almost 5 years. And today I'm presenting on what I'm researching for my master's, which is looking into the defense chemistry of Salix arizonica. So Salix arizonica was first discovered in the White Mountains in eastern Arizona and described by Robert Dorn in 1975. The species occurs within all four corner states, where the habitats are subalpine meadows occurring anywhere from 8,300 feet to 11,000 feet in elevation. The growth habit appears to be anywhere from mat forming or tall shrub from up to 3 meters tall. The key leaf characteristics for identification is that the shape ranges from elliptic to ovate with a cordate base and its glabers on both sides. For the female catkins, the capsules are glabrous, being subtended by black hairy brackets. Additionally, Salix arizonica is easily confused with Salix boothii, which has a much larger distribution and is known to hybridize with Salix arizonica. So why am I studying this species? It is a Forest Service listed species that was proposed to be listed under the Endangered Species Act in the early 90s. Since more populations were discovered within that time period and multi-state conservation agreement for the species was created, it was removed for consideration. Populations have been declining in numbers since modern began in the 1990s. However, it is still listed globally as threatened for extinction due to having a limited range and small population sizes. Preliminary data is indicating that there is less hybridization for the New Mexico and Colorado populations. Though it's unknown how this can affect potential taxonomic classification or listings, it is important to start investigating why populations are in decline. So several threats exist for the species, including alteration of hydrology, riparian degradation, and human use impact from development, roads, and recreation. Additionally, a willow rust is commonly found causing early leaf drop once it occurs on its host plant. Lastly, browsing by ungulates is the relationship I'll be investigating for my master's. To measure this, I'll be analyzing the phytochemistry of individuals throughout most of its known distribution. So I hope to accomplish looking into several objectives from my research. First, I'll be assessing the effects of deer, elk, and cattle browsing on the willow growth. Second, we'll be investigating whether historically browsed individuals have high levels of phytochemical defense compounds compared to individuals that have been historically protected. For my third objective, I'll be investigating the relationship between phytochemical defense compounds and the browsing levels incurred. Then lastly, I'll examine the plasticity of Salix arizonica defense phytochemistry by protecting individuals that have been exposed to grazing over this extent of its lifespan by constructing new protection for the individuals from ungulate browsing. Targeting populations that have high herbivory impact will help provide insights into chemical responses once stress is reduced. And here is a map where Salix arizonica occurs. The black circles are the locations that I collected from in 2022. So I'm collecting leaf tissue samples from individuals twice a year, once in the beginning of the growing season and second towards the end of the growing season. I hope to capture when browsing is most likely occurring by collecting twice in the season, whether it's early seasonal or late seasonal browsing. Additionally, I'm collecting measurements on phenology, individual sex if it's available, height and diameter of the tallest stem, whether there's protections, and lastly, browsing levels, which varies from none to heavy. Additionally, when I'm in the field, I'll mark each individual with flagging so I can resample from them later in the season, or this upcoming season. And for last season, I was able to make collections throughout its range. However, I was unable to collect from the populations twice per the growing season, mostly due to wildfires in Mexico causing forest closures. So for Arizona, Salix arizonica only occurs in the White Mountains and the eastern part of the state. Additionally, it was important to collect from populations since browsing exclosures were constructed in the 1990s. I was able to collect from 9 different populations, where the black circles indicate the populations that have the intact browsing exclosures, and the red circles indicate collections from populations without functional exclosures. Here you can see the willows are able to grow to be more robust and vigorous, but some have also been able to outgrow their exclosures, which has allowed for browsing to occur on them. In New Mexico, I was able to collect from 3 different populations, 2 which occur in wilderness areas. Since there's indication that the populations have less hybridization, I wanted to increase my collection numbers and create or fix exclosures to meet my 4th objective of looking into the plasticity of the defense chemistry. In the Carson National Forest in northern New Mexico, you can see in the foreground, in between the trees, there's an exclosure in the meadows with active grazing occurring on the left side of the photo. So for folks working at the Institute of Applied Ecology last season, we're surveying populations in New Mexico, and they graciously helped me with fixing this one particular exclosure to help keep the cattle out. Since it's too short to keep elk and deer out, I also created chicken wire cloaks, which you can barely see in this right photo within the fence to help deter any browsing from occurring. Additionally, in the San Pedro's Park Wilderness area in Santa Fe, I was able to protect several individuals with the existing T-post and hog wire that I was able to carry out. And luckily, because I did not bring enough out, I was able to find some nearby and create this fixed exclosure that is about 7 feet tall, should be sufficient in keeping deer and elk out. And here you can see that this one individual used to be protected by a simple wood fence, so I also reconstructed it with some fresh downed material and chicken wire to reinforce it. And additionally, in the Pecos Wilderness in New Mexico, I wasn't able to construct anything with T-posts, but I just used chicken wire cloaks to protect about 15 different individuals there. For Utah, I focused on collecting from 2 different populations where I felt confident with field identification. The populations in this more bottom right corner by the Boulder, Utah area looks like they were either Salix arizonica or highly hybridized individuals, and I wanted to focus my collections more on just the pure identical or identification-based Salix arizonicas. Since I wasn't sure if I could create new exclosures in the wilderness areas in New Mexico, I also created a few exclosures in the Fish Lake National Forest, where it appears there's heavy browsing. Here is the Neoch Meadow, where there's a large thicket of Arizona willow. And my coworker and I constructed these 4 exclosures that you can see, protecting about 5 different individuals. So overall, I hope that investigating the relationship of browsing effects on Salix arizonica's defense chemistry can help provide insight on whether browsing is detrimental to the species' survival or not. Whatever the results are that I do find, I hope they can provide valuable information for land managers on how they can properly manage the species. And with that, thank you for listening to my presentation.